Hello students, in today's class we will be talking about the pollen-pistil interaction. So what is pollen-pistil interaction? So just after pollination, when the pollen grain it reaches on to the surface of stigma, there is a series of events that begins so that the process of fertilization can be achieved. And that interaction that occurs between the pollen and the pistil is called as the pollen-pistil interaction. So it is after pollination when the pollen grain reaches on to the stigma, there is a series of events occur to establish fertilization, and that interaction between the pollen and pistil is called as pollen-pistil interaction. So the pollen-pistil interaction is divided into different steps. The first one is pollen recognition, that whether the pollen is the right one or not. So that power is given, has been to the stigma. Then the next is whether the stigma will accept or reject the pollen grain based on whether the pollen is compatible or non. Okay. So if the pollen grain is compatible, then there will be a process of pollen germination, and then there is pollen tube growth. So these are the different steps that occur within the pollen-pistil interaction. So first we'll talk about the pollen recognition. So now we'll talk about the pollen recognition. So stigma, it has the power to decide whether the pollen grain is the right one or the wrong one, whether to accept or reject the pollen grain. So how it does that? So when the pollen grain it reaches on to the surface of the stigma, the pollen recognition starts. So this is kind of a lock and key where the pollen grains, they have some specific proteins and glycoproteins that are present on their surface. The stigma, it also has some specific receptors. So if that matches the proteins on the surface of pollen grain and the receptors on the surface of stigma, if that is similar, if it matches like a lock and key mechanism, then it is how is going to accept the pollen grain, or else it will reject the pollen grain. Okay, so this acceptance and the rejection is based on whether the pollen grain is compatible or incompatible. So what do you mean by the compatible pollen? Means that the pollen grain is from same species but different plant. Understood? Compatible pollen means the pollen grains are from the same species but different plants. So what will happen? This will ensure that cross pollination is achieved, or you say there is cross will occur. So this is going to help in creating the genetic diversity. Now what is an incompatible pollen? So if the pollen is from different species, why this will happen? Because pollination is random. Through wind, insect or bat, anything from biotic or abiotic, different pollens can from any species can land onto the stigma. So if the pollen is from different species or same plant, it will be rejected. Okay, same plant, therefore it will prevent self. That is how the plant it encourages always a cross pollination over the self pollination. So that is how the stigma it recognizes whether it is a compatible or non-compatible pollen, and once it decides whether it is a compatible or non-compatible, if it is a compatible, the specific proteins will bind to the receptors and it will allow the next step, which is the pollen germination. So now we'll talk about if it is a compatible pollen, then what is the next step. Now we'll talk about the compatible pollen. So when a stigma recognizes that it is a compatible pollen, so what the next step happens is the pollen germination. So in that what happens? So there are different, I have made the structure of the pistil where a compatible pollen is recognized by the stigma. So there are different pollens and this one is the compatible pollen. So what happens during the pollen germination, the pollen grain, pollen will absorb the water and nutrients from the surface of stigma and it will start germinating. Soon you will see there is this formation of pollen tube. So the pollen tube, it comes out from the germ pore. It comes out from germ pore. So if you remember the structure of pollen that I have told you, in the pollen there are at some places at the outer wall, at some places there are places where the exine wall is absent, those apertures called as the germ pore, from where the pollen tube it comes out. Okay, so this pollen tube, it goes through the style and it moves towards the ovary. Now how the pollen tube, it knows the path that it has to enter inside the ovary, move towards the ovule through its micropyle. So it is a chemo growth, very important. Okay, it is a chemotropic growth. So there are chemical signals which are provided from the ovule. Okay, this is your ovule. So there are chemical signals which are provided from the ovule which guides the pollen tube for its entry inside the ovule into the embryo sac. Remember, it is the chemical signals. So it is the chemotropic growth. Okay, so the pollen tube is guided for its entry inside the ovule. So now it enters. So this is the structure of, I have made the structure of embryo sac. This end is called as the micropyle. So if the pollen tube, it enters through the micropyle, it is called as poro. It is the most common, remember, it is the most common one when the pollen tube, it enters through the micropyle. The term is porogamy. If it enters through the chalazal end, this is the chalaza, right? This is the chalaza which is opposite to the micropyle. So it is called as chalazo. If it enters through the chalazal end, it is called as chalazogamy. If it enters through the middle from the integument, it is called as mesogamy. Okay. If it enters through the micropyle, it is called as porogamy. If it enters through the integument, called as mesogamy. If it enters through the chalaza, called as chalazogamy. Mesogamy, it happens in pumpkin. Okay. Now, once it enters, so the pollen pistil interaction, this PPI, pollen pistil interaction, counts these events that occur when the pollen, it settles onto the surface of stigma. There is the pollen recognition take place. Okay, whether the pollen decide whether it is a compatible or non-compatible pollen. If it is a compatible pollen, there is pollen germination occurs. Then there is pollen tube growth. Okay, the pollen tube growth is chemotropic growth. It is guided by the chemical signals which are released from the ovule, and then it enters inside the ovule through the micropyle called as porogamy. Now we'll see what happens next. So I told you, if you remember, that in majority of the angiosperms, that is 60%, the pollen grains, they are shed at, okay, here let me write. So I'm removing, okay, I'll write here. So the pollen grains, they are shed at the two-celled stage. That means there is a vegetative cell, vegetative nuclei, and there is a generative nuclei. So what happens that when the pollen tube, it is within the style, the generative nuclei, it divides and it forms the two male gametes. So here only there is two male gametes are formed. This red structured tube, pollen tube, I have formed, and these two are the two male gametes, and this is the tube nuclei, or you say the vegetative nuclei. So the role of the tube nuclei is also to guide the pollen tube growth towards the ovule. Later this tube nuclei is also degenerated. So what is left is the two male gametes. If the pollen grain, in the rest 40%, they are shed at three-cell stage. So already there is two male gametes are already formed. But in the three-cell stage, that division of generative nuclei into two happens within the style. So when it enters through the micropyle, it already has two male gametes. Now what happens next? So this I will also cover later when I will talk about double fertilization. I am just giving you a little brief here. So what happens? This is the micropylar region. I have just made an enlarged figure of this part. Okay. So this is the filiform apparatus. There are two synergids and one egg cell. So what happens when the tube nuclei enters through the micropyle? The role of synergids and filiform is that again it releases the chemicals which helps the pollen tube to enter inside the embryo sac for further its fertilization of one of the male gamete with the egg cell. Okay. So one of the synergids, it is called as the receptive synergid. Why different? Because what happens, this synergids, it will guide it entry. This one synergid, the receptive synergid, it will guide the pollen tube to enter from this region. Soon, once it has given the signal for its entry, the role is done, it is degenerated. Why? To create a space. To create the space for the pollen tube entry, this receptive synergid will degenerate and the two male gametes are released. One of the male gamete will fuse with the egg cell and it will form the zygote. Okay. The another male gamete will fuse with the two polar nuclei present in the central cell and it will form the primary endosperm nuclei, which is triploid. Zygote is diploid. So you understand the role of receptive synergid here, that it is provides a signal for the entry of pollen tube from this region. Soon it will degenerate to create a space for its entry. Later this another synergid will also degenerate. Why? Because the role of synergid is basically to provide the chemical signals for the entry of pollen tube. If it is still present, there is a chance that another pollen tube will also enter. So to ensure that only one pollen tube it enters through the micropyle and fertilize the egg and the secondary nuclei, the two polar nuclei in the center which fuses to form the secondary. So to confirm and ensure that there is only an entry of single pollen tube, later this another synergid will also degenerate, and that is how the process of double fertilization is ensured. So this we will again study in detail, but in pollen pistil interaction, remember that the steps that are counted is when the pollen, after pollination, when the pollen it reaches onto the surface of stigma, the recognition, the germination, the pollen tube growth and its entry from the micropyle. It is all covered in the pollen pistil interaction. So in my next class, I will be talking about the process of double fertilization, which is very important. So if you like my video, please like, share and subscribe. Thank you so much.